Martin, go ahead.
Welcome to ICANX Talks, Connecting the World and the Universe. My name is Professor Martin Thu, coming to you from the middle of the United States, Iowa State University. My co-host today is Professor Miso Kim from SKU University in South Korea. For this month, we have these speakers lined up. Today, we're going to be starting with our young scientists, awardees, Professor Xianting Ding and Professor Flavia Vitale. And then next week, we'll have Professor Fulan from Australian National University, followed by Rashid Bashir, Julie from MIT, and Anthony Makos. For today, we're going to have two speakers, and we're going to be moderating. Professor Kim and I are going to be moderating, and we'll start off with Professor Xianting Ding from Shanghai Jiao University in Shanghai, China.
Okay, so I'm going to share my screen, right? So Martin, do you see my screen? Okay, so I'll be starting right now. It's my pleasure to present here, on the ICANX platform. It was such a pleasure, last year to, present together with a lot of other young scientists all over the world. I did learn a lot. And from my own lab, we did the research about single cell proteomics. So here really has two keywords. One is single cell, because we always want to use tiny amount of cell from any particular patients for diagnostics or for therapeutics, right? And the second keyword is called proteomic. In human body, we have many omics. A lot of people do genomics, they do transcriptomics, they do metabolomics, but in our lab, we only focus on proteomics. So single cell proteomics together, builds up the keywords for our research. And also we are trying to, translate our technologies to real clinical, applications. So today I'm going to share, another three, real cases where we apply our own techniques for the real clinical settings. Okay. So why do we need, single cell based proteomic, technology? So here is a simple explanation. In our human body, we have a lot of proteins. For now, people know that we have about more than 10K proteins inside each organ. So any single protein, when it overexpress or down-express, people tend to get diseased, right? So there is a request, to detect as many protein that are related to disease as possible for any particular patient. However, if you go into the hospital, you will find out that for any single patient, the amount of cells that you can provide is often very limited. Okay, for our blood body check, blood could be several vial, but for biopsy, like tissue biopsy, we can only acquire very tiny amount of cells. So there is a practical request that we need to take advantage of very tiny amount of cells, but we need to detect many proteins simultaneously with this tiny amount of cells. So this technical request actually is a global challenge, right? It's a global challenge. And this global challenge is very necessary for disease diagnostics, for disease evaluation, and for personalized therapeutics. So this is something we're trying to build up. And for conventional, single cell protein analysis approach, the most famous technology is called a fluorescent labeling, detection, or called a FACS, okay, fluorescent-based labeling. So the mechanism behind it is that if we acquire several cells, from a single patient, we want to detect many different proteins, but human eyes cannot see them, right? So what we do is that we label the target protein with a color. For example, we have two proteins that are related to disease that we want to detect. We label one protein with red color and label another protein with green color. So by detecting the intensity of the color, right, how bright the color is, this brightness corresponds to the protein expression level of that cell, right? So what people really do is to use color as a labeling tag to label the protein that you want to detect. So this approach is called a fluorescent labeling, approach. However, this fluorescent-based labeling technology has two internal limitations. The first one is color has very limited multiplicity, right? We have red, green, blue, yellow, so usually 7 to 8 different colors, let's say. So color does not permit very high multiplicity on one single cell, okay? So that's one bottleneck. The second one is light has overlap between one another. For example, if you label green color for one protein, you label blue color for another protein, and if you're trying to detect the signal at 500 nanometer detection here, so both green and blue labeling will give you some signal at this detector. So that means you use two color to label two protein. However, these two color will detect them. They overlap with each other. That you do not know how much signal comes from protein A, how much signal from protein B, right? So that makes the detection become non-specific, right? So these two bottlenecks, limited multiplicity and overlap channels, make color-based tag not very desirable, right? So facing such limitation, we have been trying to think an alternative solution. If fluorescent labeling is not desired, what else can we use? Okay, so we know that fluorescent labeling has limited amount of channel numbers. Also, the channels themselves overlap with each other. Can we find another way to label protein so that the number of channels can be increased? Meanwhile, this number of channels do not overlap with each other. Okay, so then we think of how about using elements? Okay, using elements. The benefit for using elements is, one, elements has many choices, right? So in nature, we have more than 100 different elements on the elemental table. So that means we have many choices. Second, element, they do not overlap with one another, right? For example, if you use one element to label protein A, use another element to label protein B, these two elements, they do not overlap with each other. Each element has their unique molecular weight. So therefore, we increase the multiplicity, meanwhile, we increase the specificity, right? So that's our strategy, to replace the fluorescent labeling system into elemental labeling system, to replace the fluorescent detection into the mass detection, okay, into mass spectrometry detection. So with such strategy, we develop an alternative technology platform, okay, called mass spectrometry. Okay, so if we apply this mass cytometry for single cell, then this technology becomes single cell mass cytometry. Okay, so the pipeline is, if it acquire some cells from one single patient, we can use element to label the cell surface biomarkers. Okay, these surface biomarkers distinguish one type of cell between another subtype of cell. We can also use another group of elements to label the cell intracellular signaling proteins. Okay, these intracellular signaling proteins decide the functionality of the cell. Okay, so we can use like 50, 60, 70 different elements to label both the surface protein biomarkers and the intracellular biomarkers. Then we build up something called microfluidic devices. Okay, these microfluidic devices enables us to capture one single cell and transfer one single cell into a mass detector. Okay, so this mass detector detects the element abundancy of each single cell. Therefore, one single cell protein spectrum will translate it into elemental expression spectrum. Okay, so then we can analyze how much protein A this cell has, how much protein B this cell has, so on and so forth. Therefore, we reach high dimensional, accurate protein detection on one single cell. Okay, so currently our lab okay, we can already achieve more than 50 target protein co-analysis on one single cell. So this is extremely valuable if the cells are those rare cells from a single patient. Okay, so in our lab, we focus on how to label the elements for every single cell. We call elemental labeling technology. We also focus on how to build up accurate microfluidics to ensure it's one single cell by another one single cell. We also work on mass spectrometry hardware design. And it also works on, when you get this big data, how to analyze single cell proteomics data. So we have our own bioinformatics team. Okay. And last, we're also trying to translate our technology from lab to clinical side. We have been working with a lot of clinical doctors trying to apply our techniques to solve the practical clinical questions. So I'll be introducing several cases at the end of today's talk. Okay. Meanwhile, if the cells we acquire is not in blood, it's not like this suspended cell. Sometimes the samples we acquire is not suspended cell. The samples we acquire is tissue. Okay. For example, tumor, we always acquire tissue, right? For example, the skin problem, we also acquire tissue. So we still want to examine many proteins together on one single tissue. How can we do that? So we also developed another technology called single cell imaging mass cytometry. The way we do that is very similar as the previous single cell mass cytometry. Okay, we acquire a tissue sample. We still use elements rather than fluorescent to label the proteins on each single cell, on each single tissue. Okay, so 100 different elements label different cells and different proteins. Then we use a UV laser. UV laser, the spot of a UV laser can go down to 100 nanometer, while a cell usually is in the scale of 10 to 20 micrometer, right? So the UV laser can help us to cut or to ablate one single cell away from this tissue, right? So we use UV laser as a tweezer to pick up one single cell, another single cell, another single cell, another single cell. So on and so forth, okay? So we are able to pick up every single cell on this tissue. And we then send each single cell to the spectrometry, just like last slide here, okay? We pick up each single cell and we send this single cell into this mass spectrometry detection and then do the analysis. So therefore, we can also acquire one single tissue, one single cell resolution over 50 target protein co-imaging capability, right? So now, no matter whether the samples you are providing is blood cells or tissue samples, we can both realize over 50 different protein simultaneously co-analysis and single cell resolution, okay, for every single patient's rare cell samples, okay? So to realize such kind of capability, it's a typical multidisciplinary research. So that's why I always personally very fascinated by working with different researchers from different disciplinary, because to label elements on cell, this is typical analytical chemistry research. Microfluidics is a typical mechanical engineering research. Big data analysis is typically bioinformatics research, and clinical translation, we have to be able to work with doctors. So these several, disciplinary has to be deeply collaborated. Then we can deliver such an overall technology platform into the real clinical settings. Okay, so therefore, in our lab, we have been divided our lab into 3 different groups. The first group works on how to label elements on single cell. So we call them elemental labeling system small team, okay? The second team works with algorithms. We'll get single cell proteomics, how to correlate one protein with another protein so as to deliver a better diagnostic, outcome. So we have to build up our own algorithms. So the second bunch of people, they are all mathematicians or computational biologists. And the third one, we have been working with how to validate, the target protein that we identified from the first 2 pipeline indeed can be used as the biomarker in real clinical settings. So we have to develop another technology to cross-verify the findings from the previous two. Okay, so our lab is divided into 3 subgroups. Okay, the first group works with labeling strategy, meaning how do you label elements on single cell? We call that single cell high dimensional proteomic analysis team. Okay, so in Europe or in America, people commonly use polymer, polymer to label elements into the cell. However, polymer is a very long and large linker. That means if you have one single cell and you want to label 50 different elements with 50 different polymers, each polymer will occupy some space on the surface of the cell, right? So that means if you label many polymers together on one single cell, after 30, 40, there will be no space anymore for further labeling. Okay, so we need to reduce the size of the linker. So our lab tried to change the polymer system with something called a metal organic frame, MOF, or metal organic frame. So this metal organic frame is very tiny. Okay, so it has many elements piled up in a very small volume. So therefore, we can label the single cell with much smaller space occupation. As a result, on each single cell, we can label more dimension without this traffic congestion. Meanwhile, this metal organic frame can have much more elements embedded together. And as a result, the detection sensitivity can also be increased. Okay, so that's our strategy, how to label the elements into a single cell. And also how to guarantee this linker is specific to the target protein. So if you have been working with protein analysis, you should understand that antibody is a very typical way to enhance the biological specificity. However, antibody is generally very expensive. Okay, if you work with one single protein analysis, that's fine. But if you want to detect 100 different proteins on one single cell, that means you need 100 different antibodies. Then the cost will be extremely high. Okay, so in our lab, we're also trying to replace the antibody with new biological specific probes. We call them aptamer system. So aptamer is much smaller. It's easier to fabricate and it's more stable. Okay, does not denature easily. So therefore, when we replace 100 antibodies with 100 different aptamers, and each aptamer labeled with our MOF, tags. So such system, the overall cost will be much lower than conventional antibody plus polymer system. Okay, and a lot of people say aptamer, because it's so small, how do you guarantee the specificity to the target protein? Is it durable? So our strategy is that we adopt computational biology. Okay, so rather than using a linear aptamer, we use carefully designed structure, meaning this aptamer, when it loads on the surface of the cell, it has a certain hairpin structure. And also we try to put some modifications on this aptamer to better enhance the spatial recognition between our probe and the target protein. So therefore, without sacrificing the specificity, okay, the aptamer can be loaded onto the single cell. So now you can, when you review our elemental labeling system, you will realize that, yeah, we have our own linker. We replace antibody with aptamer. We design our aptamer into certain structure so to increase the specificity. Okay, so we have done a lot of work, in this domain. So the second group works with data. So after we acquire so-called single cell proteomics data, we need to find out a way to analyze that, right? So say if we can detect 100 proteins on one single cell, that means each single cell will be outputted by a vector of 100 proteins. how to analyze this protein-protein interaction then becomes a very big headache. Okay, so our strategy is that we multiply this 100 protein vector with 1,000 random matrix to further project this 100 protein correlation into a much higher space. Okay, so if in lower space, protein-protein interaction is very strong, then if you multiply by 1,000 random matrix in higher domain, the interaction is still there, right? However, if in low dimension, this protein-protein interaction is very sparse. That means if you project them into a much higher space, then the chances that they are still together will be very low, right? So in much higher space, we run something called a winner-take-all strategy. We set up a threshold. Okay, any correlation above this threshold, we define a strong correlation. Any number below this threshold, we define very weak correlation. Therefore, okay, we rerun this dimension concentration to do cell-cell subcluster differentiation based on their protein expression panel. Okay, then we can get much better, rare cell subtype distinguish. So when we apply our algorithm to the real, clinical data, we can see that two typical algorithms, they do not distinguish those rare cell subpopulations very well. With our data analytical strategy, we can distinguish those rare cell population much clearer compared to the standard algorithms. So when we apply such algorithms into the real disease cohort study, that means we can identify those disease-relevant rare cell subtypes with much higher confidence. Okay, and then we compare our algorithm with all the other mainstream algorithms in the world. So based on cell number, protein number, cohort size, protein expression intensity, we build up something called an algorithm decision tree. Okay, so this algorithm decision tree maximizes the statistical confidence for any given data frame. So that means when a non-biostatistician, when a first-line doctor acquire their data, they do not need to understand the biostatistics anymore. They just need to simply input their data frame into our pipeline. Okay, our pipeline will automatically decide the best statistical algorithm to analyze their data and output the results with the highest statistical confidence so that they can make decision based on this data analysis to treat their own patients. Okay, so I think this solves a very important, bridge between the engineering lab and the first-line clinical doctors. And then we also hired another PhD. She was working with Google before, and we try to analyze something called a cell cluster association network, which means is that we do not focus on any one single cell subtype. We are trying to build up cell-cell connection network. This is like each cell subtype is a human person. Another cell subtype is another human person. Okay, if you only use one human person as a diagnostic biomarker for complex disease, usually it's not enough, right? If you examine the human-human interaction pattern, okay, this interaction pattern, what we call the topological network, much better describe the social behavior of the society, right? The same thing for any complex disease. Rather than using one single cell subtype, we use cell-cell internal connection to decode the mechanism of the disease with much higher confidence. So basically, we borrow the algorithm from Google. We borrow the algorithm from Facebook. Okay, so they use the similar algorithm to study human-human interaction society. And we borrow the same algorithm to study cell-cell subtype communication network. And we construct the very complex cell-cell interaction network and use this network dynamic change to describe a complex disease. So that's what we do, okay? So the third group works with target validation, which means if you understand, oh, this protein and this protein, they are very correlated with the disease progression. Then for real clinical use, we need to detect them at very low dose with very small number of cells, right? So to verify whether we can do this, we build up a new technology called hydrogel, or called photoclick hydrogel. So this photoclick hydrogel material has two superior properties. The first one is a mixture of proteins loaded on this hydrogel under the electrophoresis. Okay, the protein can be quickly separated based on their charge to molecular weight ratio. Okay, for those proteins who have higher charge with lower molecular weight under the electrophoresis, they will run very fast. But the other proteins which lower electro loading but higher molecular weight under the electrophoresis, they will run very slow, right? So based on charge to mass ratio, they will be quickly separated under the electrophoresis inside the gel. And then we use the UV light to shine on this gel. Okay, this hydrogel material, they will absorb the UV light energy. And then they will have chemical reaction with all the proteins inside the gel. Then this protein will be anchored, will be covalent bonded inside the gel and do not move anymore. Then we can cut the corresponding protein slide and detect how many protein molecules there. So therefore, we can separate the proteins from a mixture of protein, fix the protein inside the gel, and detect the protein with much higher accuracy. So this hydrogel technique enables us to detect very low amount of protein very accurately on one single cell. Okay, so here is the comparison. Using our gel, which called the MMP gel. For every one single cell, we can detect very low amount of protein very clearly. Okay, and furthermore, we expand this one single cell detection platform into a multiplex single cell detection platform by using MEMS technology. Okay, so we fabricate a lot of single cell well on one wafer. Okay, so each single cell well, the diameter is about 15 micrometer to allow only one single cell loaded into this gel, into this well. Okay, so on the same chip, we have about 10K single cell wells. That means we can simultaneously load almost 10K single cells on one chip. Okay, then we use the first well and the second well. Between them, we loaded those photosensitive gel as mentioned in last slide. So therefore, the first cell lysed and all the protein from the first cell will be detected in the first channel. Okay, and the second well has another cell lysed, and the proteins in the second cell will be analyzed in the second channel, so on and so forth. So now we can achieve almost 10K cell simultaneously loading and detection at the same time. So this MEMS chip further increase the throughput of our technology. Okay, so now we can simultaneously work with 10K cell and one single cell, very low amount of protein, quantitative detection. Okay, so then this chip permits in the real one single drop of blood, many cell co-analysis capability. Okay, and furthermore, we develop something called a fluorescent and mass dual tag system. Okay, so the labeling linker now has both fluorescent color and elemental signal. We can use fluorescent color to sort the desired cells from many cells. Okay, so most of the time, the desired cells that we want to detect is only small percent for the whole cell population. So using fluorescent, we can quickly sorting the target cells outside from the main population. And then we can use our mass signal to detect the protein with much higher, abundancy and of confidence. So this fluorescent plus mass dual signal tag will cross-verify each other and achieve both high resolution and high multiplex imaging when we apply such labeling system on this single cell imaging mass cytometry, right? Okay, so after we build up all this technology, we have been collaborating quite a lot with our hospitals to run clinical translational applications. So in the past 10 years, we have been working with almost all the major organ, disease, from blood to skin to liver to heart. Okay, so for any biopsy sample, which means very tiny amount of tissue or very tiny amount of cells, if you want to do personalized diagnostics, we offer a very, robust technology platform. So for time issue, I will only give you 3 cases to demonstrate how to use such kind of technology. Okay, so the benefit of our technology is we can acquire very super large personal data using very trace amount of cells. So for example, if you want to examine 30 different cell subtypes for 30 different functions, like heart, liver, brain, okay, for cell division, apoptosis, necrosis, proliferation, okay, 30 cell subtype, 30 different function, okay. If you use green, blue to 7 color fluorescent labeling system, you need 100 times to acquire such data. So even if you use 14 different label system, you still need to do 18 times. But for our technology, you only need to acquire 1 single sampling. You can acquire such big data. So that offers the clinical side a much better data presence, right? So 3 cases, okay, to show how we use our technology. So the first case is to identify new biomarkers. So in this case, we are interested in stroke. Stroke is a worldwide challenge without an effective biomarker in blood. Stroke, whenever it occurs, you need very tight time window to cure this patient. So if the diagnosis of stroke can be much earlier than the occurrence of the phenotype, then we can save a lot more patients. Okay, so our hypothesis is when stroke happens or before the stroke happens, the blood-brain barrier inside this patient already starts to have some cracks. So that means some cells that originally only belong to brain may have a chance to bypass this blood-brain barrier and then go into the blood system and circulates around your body, right? So that means even before the stroke happens, there could be some cells that only belong to brain that already show up in your blood circulation system, right? If we can detect the number of such cells, these cells may be served as a super early diagnostic biomarkers for stroke, right? So that's the hypothesis. And we use our technology to do this, okay? First, we have our animal model, okay? So we establish this stroke animal model. You can see the healthy mice, their brain are very, healthy, right? For the stroked mice, their brain, because lack of blood, right? Their brain become very white. So after the model, we use our technology to screen which type of cells we can detect in blood circulation system. We found out some cells that co-express these 3 protein biomarkers show up in their blood circulation system. One of the marker is neuron marker, okay? Neuron marker is a very typical marker for neuron. And these neuron cells shall only exist inside the brain and shall not exist inside the blood circulation system, right? So then we believe this circulating neuron cells may be served as the stroke diagnosed biomarkers. So whether this is true or not, we further move on for clinical settings. So we recruit 63 patients with different disease, with different degree of stroke, okay? And we again analyze the so-called circulating neuron cells inside these patients, and find out that in patients, the conclusion is the same as in animal. Okay, the healthy patients, in their blood, they rarely show circulating neuron cells. However, in the stroked patients, their blood has more or less circulating neuron cells. Okay, so I think this is very important because circulating neuron cells then may be served as early diagnostic biomarkers for stroke, which is extremely meaningful. Okay, that means we can save a lot of lives even before the stroke occurs. The second case, we are interested in organ transplantation. Okay, tacrolimus and rapamycin are the two first-line drugs that treat organ transplantation repellation. Okay, so when a patient receives a new organ, they may repel this organ. So these two drugs are used to cure this phenomenon. So we recruited 140 patients. We are trying to understand which drug shall be chosen for personalized organ transplantation. Okay, we follow the two groups of patients using different therapy for up to 3 years. And we find out that for the first drug user, their T cells become very old. Okay, that means this drug may tend to make their T cells very tired and become old quickly. While for another drug, the regulatory T cells become elevated. Okay, so then we reach a hypothesis that long-term intake of the first drug, because their T cells become old, so at their later age, the risk of developing infection and tumor should be higher than this group. Is that true? So we stimulate these patients with another stimulation, and we found out that the T cells in this drug treatment group, the capability also becomes strong. Okay, so our hypothesis that using this drug at their later stage, they may be healthier than this group. Okay, then we really do this study and recruit another 100 patients and examine their later life and find out that yes, the user of the first drug apparently has much higher chances to develop infection tumor compared to the second one. So therefore, this technology offers a new way for personalized medication choice. Okay, so the last case, we are interested in SLE disease. So SLE disease is a typical complex autoimmune disease. Okay, and the mechanism is so complicated that even till today, nobody knows why a person developed SLE. So we use our technology to examine all the T cell lineages inside the patients, especially for T helper cells and toxic T cells. We examine all the different subtypes and try to identify which one or which several of T cell subpopulation they work together, form a network, and lead to the disease. Okay, so here each dot, every single dot means one cell subtype. So millions of cell subtypes are screened, and we can apparently see that in healthy control and the prices, these two groups, at least 3 different cell populations are abnormally expressed, like 4, 3, and 2, right? So in healthy control, people do not have them, but in the diseased patients, these cells become abnormally increased. So these 3 cells are listed down there. So the later 2 has already been reported by the other researchers, but the first one, this cell subtype has never been reported that is correlated with autoimmune disease. Okay, so we use another typical way, they call the immunostaining, to cross-verify, yes, this cell subtype is indeed abnormally expressed. And then we recruit another group to verify that these cells, not only at skin tissue, but also at the blood system, abnormally increased. So therefore, we kind of understand, oh yeah, so this cell, together with the other 2 cells that has been previously reported, they work together, form a network, okay, form a group, and damage the whole immune of the patients so that the people become diseased. Okay, so in the past 10 years, we have been collaborating with all the researchers all over the world. Okay, and we have been collaborating with molecular biologists, biostatisticians, medical doctors, and mathematicians. So it's really fun, to collaborate with people from different interdisciplinary. So that's why I always encourage the younger generation, the young students, try to collaborate as much as possible with people from these different interdisciplinary. For the time issue, that's all I want to talk for today. Okay, I want to express my special thanks to my PhD supervisor, Professor Qiming Ho, for the consistent help, and all the other collaborators, also my lab mates, for all the good work. And our lab is has a postdoc opening, so if you want to do similar research, feel free to contact me. Okay, thank you for all. That's all for my talk. Martin, I'm giving my floor back to you.
Thank you.
Okay, thank you very much, Xianting. Martin, would you want to say something?
No, it's your turn.
Yeah, okay. So we will talk more about this proteomics and then the clinical translational applications more after Flavia's talk. So hi everybody, my name is Miso. I'm an assistant professor. I'm a co-moderator for this great talk show. And then I take the privilege to introduce our next speaker, Professor Flavia Vitale at the University of Pennsylvania, Philadelphia. So like Xianting, she belongs to the first cohort of our young scientists, ICANX Young Scientists awardees, who are all of them are very like the super rising stars. And it is my great pleasure to introduce her. Flavia, it's yours. The word is yours here.
Thank you. Thank you so much, Miso, for the introduction. It's my great pleasure and honor to have the opportunity here to come back on the global science stage of ICANX. Can you see my slides well?
Yes, very nicely.
Great, thank you so much. Yeah, so I was honored and privileged to be awarded to be part of the first cohort of the ICANX Young Scientists. And today I have the pleasure to come back here and tell you about our work on engineering high-resolution multiscale bioelectronic technologies based on nanoscale soft conductors, and in particular our research work on Maxim bioelectronics. So bioelectronic technologies are powerful tools to address the societal and economic challenges posed by a variety of neurological and neuromuscular disorders. Typical bioelectronic technologies are, for example, implantable brain stimulators to treat diseases like Parkinson's or epilepsy, implantable spinal cord stimulators to treat chronic pain, and cochlear implants for restoration of auditory function. All of these technologies I just mentioned are have been a mainstay in clinical neurology for decades. There are a number of 100,000 patients who have been implanted and live with these devices. Other type of bioelectronic technologies like brain-computer interfaces for restoration of paralysis or control of active prosthetics hold the great promise of restoring function, but they are still in the research realm. So in my lab at the University of Pennsylvania, we are developing novel bioelectronic technologies to study, diagnose, and treat neurological and neuromuscular disorders. Our ultimate goal and mission is to translate these technologies to the hospital and ultimately improve patient lives. To accomplish this goal, we adopt a multidisciplinary approach, combining fundamental studies of nanomaterial properties at the interface with biological systems, with the development of custom manufacturing processes to integrate these materials into functional devices. And we then use our devices to research neural function and disease in a number of models, ranging from small animal models of neurological disorders all the way up to humans. Some of the examples of the technologies that we developed in my lab and that I am going to tell you about today include wearable and implantable sensors for clinical monitoring and diagnostics in humans, as well as multimodal neural interfaces for basic science studies, so transparent optoelectronic devices. At their core, all of the bioelectronic technologies rely on an interface between man-made electronics and biology to transduce and control bioelectrical activity. Despite years of research, the mismatch between the physical, chemical, and electrical properties of biological systems and artificial components is still a significant challenge that severely impacts the performance of bioelectronic devices. Biological tissues are soft and compliant. They are dynamic and moving. Think about how your heart expands and pulses at every beat. Bioelectric signals are conveyed through ionic currents in fluid, and all materials are organic. Traditional bioelectronics, on the other hand, are rigid and static. Signal in the solid conductors are carried through flow of electrons or holes. And these technologies rely on inorganic materials like silicon and metals. And also manufacturing these devices is typically time-consuming, low throughput, and quite expensive because the manufacturing processes still rely and have been directly translated from traditional microelectronics and MEMS. In recent years, nanostructure carbon-based nanomaterials like carbon nanotubes, graphene, nanodiamond, and MXenes have emerged as promising alternative to metals for safe and long-term stable bioelectronics. The main advantages of nanocarbons are in the broad range of properties such as the high electrical conductivity, the high surface to volume ratio, the low density, and the magnetic properties, in particular, the low magnetic susceptibility mismatch with soft tissues. These materials can also be easily functionalized to enhance or add sensing modalities, like, for example, to make enzymatic detectors. They have generally high photothermal conversion efficiency, which is favorable for use, for example, in remote control of neural function via photothermal stimulation. In addition to the peculiar material advantages, carbon-based nanomaterials can be easily processed into the final devices with high throughput, low-cost manufacturing, such as inkjet printing or direct laser writing. And today, there have been a number of technologies for bioelectronic interface that have been proposed and validated and are based on carbon nanomaterials. They include implantable brain electrodes, peripheral nerve recording and simulating electrodes, wearable sensors, as well as electrochemical biosensors. In the last few years, in my lab, we have been extensively investigating carbide MXenes for bioelectronics. The term MXenes broadly refer to two-dimensional order structures consisting in single to 3 atomic layers of an early transition metal, a conductive nitride or carbide core, and a surface decorated with oxygen, hydroxyl, or fluoride groups, depending on the synthesis process. And MXenes are typically synthesized from wet etching, selective wet etching of the A atomic layers, typically aluminum, in layered ternary carbides and nitride precursors. They are also called max phases. And etching of the A phases is typically conductive in aqueous solutions of hydrofluoric acid, followed by intercalation and mild sonication to delaminate MXenes and produce single flake suspensions. Of the more of the 40 forms of MXenes that have been synthesized so far, we have been primarily focusing on titanium carbide MXenes, since it is the most extensively characterized and the best performing at the moment in terms of conductivity and capacitance. Now, what makes titanium carbide MXenes unique among other carbon-based nanomaterials like carbon nanotubes and graphene is that in addition to all these properties I just mentioned, the repulsive forces between the negatively charged two-dimensional MXene flakes make the material stable and in aqueous suspension, which means that we can use MXene directly in a form of aqueous inks without any additional functionalization. The processing of MXenes in liquid phase is then safe, scalable, and room temperature, and compatible with common polymers used for encapsulating bioelectronic devices. Since titanium carbide MXenes is a relatively new material with little or no precedent in bioelectronics, in the early stage of our work, we really put significant effort into developing a fabrication process to reliably produce bioelectronic devices based on MXene. And this process is a typical, consisting typical typical lithographic and micropatterning steps integrated with spin coating the MXene ink onto the flexible polymeric substrates, typically perylene, to micropatterns the contact sites. Using this process, we successfully produced microelectrode arrays, so devices, thin film devices with microscale features with different geometries. An example of one geometry is an all MXene planar array that is designed to sit on the surface of the brain underneath the scalp, consisting in 50 micron electrodes spaced at 300 microns apart. The second configuration is a penetrating electrode, so an electrode that goes inside the brain, made with two adjacent columns spaced 35 microns apart. The first column is composed by a set of 25 microns MXene contact, and the second one is composed by same size gold contact. And we designed the structure to conduct side-by-side comparison of the recording performance between MXene and the gold standard, no pun intended, inside the brain. Now, before I move forward, let me take a moment to briefly discuss the mechanisms governing the generation and transmission of electrical signals in the body and how this can be sensed and modulated by a solid conductor, because this concept will return several times throughout my talk. So inside the body, the bioelectricity is the result of the flow of charged species in an aqueous electrolyte. So basically flow of ions. For example, in the brain, the signals are generated by flows of ions in and out the neuronal membranes, and then that are then propagated through a network of interconnected neurons. When a solid conductor is placed inside the extracellular space at the solid-liquid boundary, ions will self-organize into a layer structure consisting in a solvated ions and co-ions immediately in contact with the electrode surface, and then ions accumulated further away in the diffuse layer. The charged electrode surface and the solvated ions in the center layer will act as a half plate of a capacitor. And so current is capacitively transduced from ionic flows in the electrolyte or in the extracellular space to electron flow inside of the conductor. There's also an alternative charge transfer mechanism, which is directly flow of electrons from metals that can undergo that are multivalent states or pseudocapacitors. Now, depending on the flow of charges, so from the metal to the electrolyte medium, then the electrode is stimulating. If charges go from the electrolytic medium to the electrodes, then the electrode is recording. So in analogy with electrical circuits, the ability of an electrode electrode to transduce ionic currents can be characterized by its electrochemical impedance. So the impedance at the interface between the electrode and the electrolyte. With electrochemical impedance spectroscopy in saline solution, which approximates the composition and conductivity of extracellular fluids, we compared the impedance of gold and MXene of the same size. And we found a consistent 4 times lower impedance of the MXene contacts at the reference frequency of 1 kilohertz. Looking at the phase diagram, we see that MXene exhibits a non-ideal capacitive-like charge transfer mechanism. But to further investigate the nature of the MXene electrochemical interface, and so the nature of the charge transfer across the MXene interface, we took a closer look at the electrode morphology, and then we modeled the electrochemical behavior with an equivalent electrical circuit model. The electrode surface of the MXene is formed by tightly overlapping flakes with an average lateral size of about 1.4 microns and a few nanometer peaks, which results in an excess surface area available at the interface compared to smooth metal surfaces. From fitting the impedance spectra with an equivalent circus model of this porous and rough MXene surface, we found that the charge transfer is indeed primarily capacitive, and the MXene double layer capacitance, so the capacitance of the MXene interface, is about 300 times higher than that of gold. And also functional groups, it's possible that functional groups on the MXene surface contribute and further interact with the ionic species, thus further enhancing the electrochemical double layer at the MXene interface. When we tested then these electrodes in vivo and we implanted in small animal models to acquire brain signals and then compare side by side the quality of the recordings acquired with MXene and gold, we found that on MXene contacts, we were able to detect a much larger number of neural signals, which correspond to these small negative very fast deflections here on shown on the top right. And we also found lower noise and overall improvement of the signal to noise ratio, which demonstrates that MXenes, because of the higher surface area and the lower impedance, have a much higher sensitivity than smooth metal surfaces to detect neural signals. Now, as I mentioned, MXene and titanium carbide MXene had never been used before in bioelectronics and neuroelectronics. So we also needed to explore and test the biocompatibility of MXenes to neurons. And neurons are particularly good cells for this type of studies because they are highly sensitive to their local microenvironments and to surface interaction. And so they tend to die very easily if the surface and the materials are not biocompatible. So we grew neurons for 7 days on MXene films, like those deposited on our electrodes. And then we had the control of neurons grown on standard tissue culture materials. And after 7 days, we found that there was no significant difference between the number of healthy neurons on the MXene surface and on the tissue culture plate surface. What was also interesting by fluorescent staining is that we found that the neurons could grow and form interconnected networks on the MXene surface, which means that MXenes can not only support viability of neurons, but they can actually support their growth and formation of interconnected networks, which is also very interesting for application of MXenes in neuronal tissue engineering. Now, devices that are fabricated in the clean room are ideal for applications such as recording and stimulation in small rodent brains. But really, they do not scale well for applications that require large area coverage, such as wearable sensors for humans or implantable electrodes for the human brain. And this is because microfabrication is time-consuming, is expensive, and is optimized for patterning the device features at the micro scale, but really doesn't scale well beyond the millimeter feature size, and is definitely very challenging to pattern centimeter scale features on thin film polymeric substrates using microfabrication. So to scale up Maxim bioelectronics for large area coverage, we devised, and my postdoc at the time, Dr. Nikki Driscoll, devised a rapid, high throughput and high yield fabrication process that is still based on Maxim aqueous inks. The process involves laser machining an absorbent substrate to create the electrode contacts, followed by inking with Maxim. The device is then encapsulated with a thin layer of a silicon elastomer, typically PDMS or EcoFlex, followed by selective etching to open the electrode contacts. And this process produces planar structures that can be used, for example, for skin-based and large brain sensors. Now, in some application, like non-invasive brain monitoring or electroencephalography, EEG, the electrodes are placed on the scalp. And so they have to overcome the hair barrier to make contact with the scalp and record brain signals with an acceptable signal to noise ratio. For this application, for application in scalp EEG, we slightly modified the process to add three-dimensional mini pillars at the contact sites. And these mini pillars are made with cellulose foams infiltrated with Maxims. And then all the rest of the fabrication process is similar to that of the planar electrode. So in addition to being significantly faster than microfabrication, this process is high throughput, low cost, and scalable, as it can be conducted at benchtop in any laboratory setting and without the need of specialized material or equipment. It also allows to fully customize the device design and geometry for the specific area of application. And with this process, we can produce large-scale devices, so tens of centimeter scale devices with the desired density of contacts and area coverage. Of course, in this work, we had to conduct a thorough optimization of the processing parameters to really maximize the electrical and mechanical properties of these devices. So we explored, for example, the range of Maxim concentrations to identify the percolation threshold to achieve an interconnected conductive network on the cellulose substrate. And we find that at the 20 milligram per ml of Maxim, so approximately 2 weight percent, there is no further improvement in the conductivity of the material, and that the percolation threshold is about 0.5 milligram per ml. We also explored the effect of the cellulose of the substrate on the conductivity of these constructs, and we, both with natural and synthetic materials, we achieved very good conductivity. Probably the best was with a blend of cellulose and polyester. We also tested the possibility of using the same process, right, to make devices out of other aqueous inks like organic polymers like PDOT and reduced graphene oxide. And yes, this process is amenable for to be used with other conductive inks. However, with Maxims, we achieved, because of the higher conductivity of the material, we achieved the best performance in terms of resistivity. And finally, we currently, the resolution limit of the process is approximately 250 microns, and is primarily dictated by the resolution of the laser cutting onto the absorbent substrate, but we are also investigating processes that allow us to push the resolution further down. Now, the resulting electrodes, and this is an example of electrodes with 3D features for scalpel electroencephalography. So these resulting electrodes are composed, as you can see, by conductive MXene cellulose traces and soft porous MXene cellulose aerogels at the scalp contact sites. The close-up images of the array surface showed MXene flakes well interpenetrated into the entangled substrate structure. And this is also shown by the prominent characteristic MXene peaks in the XRD and Raman spectra. And the presence of the characteristic MXene peaks in the Raman and XRD spectra also demonstrate that the process is preserving of the material, and the material does not degrade during the fabrication of our devices. We then, of course, benchmarked our MXene-based large-scale electrodes against commercial clinical electrodes made with platinum and measure the electrochemical properties that are relevant in the context of recording, so the electrochemical impedance, but also in the context of stimulation. And so we measure the ability to charge and deliver, to store and deliver charge at the electrode interface. In terms of the impedance, we found over 20 times improvement in the electrochemical interfacial impedance and more than 30 times higher charge storage capacity, which is the ability to store charge at the equilibrium at the electrode interface at the MXene interface compared to platinum, which implies an improved ability of these electrodes to store charge for safe and effective stimulation. And again, we attribute this result to the higher surface area and the highly capacitive nature of the MXene interface. From the wider water window, so the safety limit at which the electrode, safety voltage limit at which electrode can be pulsed without inducing water electrolysis and hydrogen evolution. And from the significantly smaller maximum cathodic excursions in voltage transient experiments, we found that MXene displays approximately 10 times larger charge injection capacity, which represents the ability of the electrodes to deliver charge during fast pulsing that is characteristic in bioelectrical stimulation. This 10 times higher charge injection capacity compared to platinum, and comparatively or slightly lower than best performing stimulating electrodes based on organic polymers and porous graphene. We also measured the impedance on the skin for both flat and three-dimensional MXene electrodes with 3 millimeter contact sites and compare with approximately 3 times larger commercial electrodes made with silver chloride and an electrolytic gel. And what we found is that at the reference frequency of 1 kilohertz, the impedance of MXene is comparable to that of larger commercial gel electrodes, and even more striking in the 1 to 100 frequency band, 100 hertz frequency band, which is characteristic of bioelectrical signals that are detectable on the skin, like brain and muscle signals. The impedance of both types of MXene electrodes is lower to that of commercial silver chloride electrodes by a factor of 10. For stimulating electrodes, the electrochemical stability under repeated pulsing is of paramount importance. And so we subjected the electrodes to accelerated electrical aging tests with 1,000 cycles of controlled current stimulation pulses at the current amplitude of 1.2 milliamps. And we did not see any change in the impedance behavior of the electrodes, confirming that these electrodes are stable under continuous current pulsing. We also tested the mechanical stability of these electrodes under repeated banding deformation, and we did not observe any failure of the devices after 1,000 cycles of banding at the 2.5 millimeter banding radius and the negligible change in resistance. And we also test the stability of the structure structures under mechanical deformation, bending deformation when we use different substrates, we found minimal effect from the type of structures, again, also the mechanical performance, but we will also investigate this further. In addition to the favorable electrochemical behavior, the physical properties of MXenes, of Ti3C2 MXene, make it safe and compatible with clinical imaging, such as computerized tomography and magnetic resonance imaging. Now, we knew when we started to work with MXene that its density is more than 5 times lower than that of metals like platinum, which results in almost no artifact from X-ray scattering when imaged in the CT scan. And so these artifacts appear very clearly around the platinum electrode as this like bright strikes around the electrode surface. What we didn't know, and what we demonstrated for the first time in the study, is that MXene is weakly paramagnetic with a magnetic susceptibility that is much closer to that of human tissues than metals like platinum. And we demonstrated that with measurements, magnetization measurements of the MXene electrodes. As a result, then when we imaged these MXene contacts under high magnetic field strengths at 9.4 Tesla, we did not see any imaging artifacts that were instead very clear and present, as also distortions around the platinum electrodes. We also tested the compatibility of MXene electrodes under functional magnetic resonance imaging, or fMRI, which has even higher or more stringent limitation in terms of susceptibility artifacts. So in the studies, we had a healthy volunteer with the electrodes placed on their forehead. And here are I drew the outline of the electrodes on the forehead of the participant, because actually the electrodes are almost invisible under the fMRI. And this is again a demonstration of the magnetic compatibility of MXene. We also measured the temperature during this experiment, and we did not see temperature increase, which also shows that MXene is safe in the MRI and that does not induce RF heating. Okay, so now we've developed this great technology. What are we going to do with it? And so here in the last part of my talk, I'm going to tell you a little bit about the applications of these technologies and the work that we have been doing to demonstrate the use of these devices in different bioelectronic sensing technologies. So to demonstrate the clinical utility of this MXene electrodes, the first thing that we did was to test their use in gel-free EEG. So typically, and we then benchmarked MXene electrodes against clinical electrodes. Clinical electrodes are typically made with metals, and they require a gel underneath the metal surface and above the scalp to overcome the hair barrier and make contact with the scalp, and again record brain activity non-invasively with good signal quality. The main drawbacks of these gels is that they are highly irritating, uncomfortable for the patients. They leave like a lot of residues. And in this test, so we decided, based on our findings, that MXene impedance was favorable compared to gel electrodes. We place on the back of the head of the participant the MXene electrodes without any gel underneath. And then at the center, a clinical gel electrode to, again, compare side by side the recording performance. So we found that the electrode scalp impedance of the MXene contacts is comparable with that of the gel electrodes, again, without any gel underneath. And the brain signal recorded with the two types of electrodes is virtually indistinguishable, not only at the representative selected location, but actually across the entire array. Now, given the high sensitivity and the resolution of these MXene electrodes, we can start pushing the boundaries of brain recordings. And for example, to test, the ability of recording brain activity non-invasively at high spatial and temporal resolution, we made grids of 16 MXene electrodes covering an area of 2 by 2 centimeters, which is a higher density configuration than current EEG systems. And we recorded brain activity in this case on the forehead. So now here I'm showing the okay, so we have an issue with the video, but we recorded the brain activity with a very higher spatial and temporal resolution, and apology for the issues with the video. But our results showed that we could detect brain activity at sub-centimeter scale with a density of electrodes that surpasses systems that are currently available. We also tested the MXene electrode technologies in other applications in epidermal electronics, such as high-density mapping of muscle activation with surface electromyography. And here we place an array, a grid of 40 channels of 3 millimeter MXene contacts on the biceps. And we, what we did is that we stimulated the nerve that controls the bicep muscle. And we recorded the signal generated by the muscle activation due to the nerve stimulation. So here's an example of one of the signals detected on one of the MXene electrodes. So the blue square represents the stimulation signal, the purple dot represents the muscle response. And so we mapped the latency, so the time difference between the stimulation and the muscle response. And it is very well known that in the area where the latency of the muscle response is minimum, that is the area where the nerve meets the muscle. So we found an area approximately perpendicular to the muscle length of minimum latency. And we confirmed, based also what it was known from previous studies, that that was the area of the muscle innervation. And so this is an important information because typically innervation zone detection is done with invasive or electrodes or repeated EMG sensing studies, and the ability to map muscle anatomy very effectively and high resolution with a single grid really can enable new applications in rehabilitation as well as in chemical denervation studies for plasticity treatment. And we were also able to do the same innervation zone identification also in smaller muscles, like the muscle that controls thumb abduction. Now, one thing that is like very common for the electrodes, especially gel-free electrodes, is the susceptibility to artifacts induced by motion. And so we tested the motion-induced artifact during muscle contraction as well as just like arm movement, and compare the artifacts induced on commercial electrodes. And what we found is that the artifacts detected on the MXene electrodes are actually lower than the artifacts on the commercial electrodes, and then they can be easily removed by simple filtering. We also confirmed that we could detect heart activity with the MXene electrodes with quality comparable to that of commercial electrodes. And then finally, we also implanted during brain surgeries these electrodes onto large animal models of traumatic brain injury. And we were able to map the propagation and the distribution of characteristic signals in these animals. And now we are collaborating with like several investigators at Penn and at other institutions to monitor traumatic brain injury evolution chronically in different animal models using our technologies. So in conclusion, I also want to briefly mention that while we, like my lab, is pioneering the use of MXene for bioelectronics, we are also collaborating with a wide network of investigators in US and worldwide that are interested in exploring the use of MXene for different biomedical applications, like, for example, remote neuromodulation, as well as cancer theranostics and antiviral application, which is very timely in this COVID times. And so finally, in conclusion, I just would like to acknowledge my talented group of students and postdocs. And without them, really, none of this work would have been possible. I also want to acknowledge our collaborators at University of Pennsylvania and outside, especially Dr. Yuri Gogazzi and Dr. John Medaglia at Drexel, our funding sources, and all of you for the attention. And thank again, ICANX for the opportunity to present my work here today. Thank you.
All right, thank you very much, Professor Vitale, for your very nice talk. And we're gonna get into the Q&A. We got a few questions from the audience. The first one goes to Professor Ding from Juan from Beijing. Very nice talk, Professor Ding. I'm wondering your 10K microwell array on MEMS chip, do you need to do additional surface modification before you use them?
Thanks for the question. No, actually. So what we do is that we use typical MEMS fabrication technology to fabricate a mold. First, and then we directly pour our hydrogel material on the top of this mold, and we carefully dry the material into hydrogel, and we peel it off, and then reattach this microwell that's made of our hydrogel material, carefully bonded onto a glass slide. So that's our basic process of fabrication. So we do not treat our surface with any additional modification.
Okay. Okay, thank you very much. The next question also for you, Professor Ding, for the personal precision medicine research. Do you have any real cases with patients? And do you have any feedback, and how much would it cost to personalize?
That's indeed also a very common question that's being asked from a lot of my colleagues as well. Yes, we have been working very closely with our affiliated hospital. Our university had 13 affiliated hospitals, so we have very, very big patient resource, and especially for tumor, every single tumor patient, he or herself is a unique patient. So the treatment or the combinatorial drug shall be different. So we use our technology to analyze the tumor cell subtype profile, and suggest to our clinical doctors how to prepare the personalized combinatorial therapy to treat their patients. So that's how we translate our technology for precision medicine. So yeah, the first question is yes, we do real cases quite a lot, and how much it costs. This question, I don't know how to answer that, because we do not charge our patients at this moment. We have not officially acquired FDA approval to sell this technology yet. So we collaborate on the research basis with our hospital doctors, purely for research purposes. So we do not charge. Later on, if this technology becomes commercially available, I think probably per patient, this is around like a several thousand RMB, which is several like 700 to 800 US dollars. I would expect that is the cost per patient. Yeah.
Thank you very much. Very interesting sciences. As you can see, we wish you all the greatest success. We need that kind of technology in our hospitals.
Thank you.
The next question is for Professor Flavia Vitale, and this is from Aris. Great talk. I have a question for your MX trodes dimensions. Can you move it down to micro or nanoscale, or can you navigate up and down the scales? And does that improve performance?
Yeah, this is a great question. And I think it really, the performance is dictated, the performance benchmarks are dictated by the application. And so for epidermal electronics, wearable sensors, it is, we now know that below the millimeter scale, there's really no much more information that can be gained. The resolution of the activity that can be detected on the skin is limited. But if we could move, and that's what we're working on, the resolution of these electrodes to microscale, then this would open up very exciting opportunities for implantable electrodes, primarily for brain and peripheral nerve recordings, because we could then be able to interface and detect with activity at the single cell level. And this is like very important, particularly for example, applications in brain-computer interfaces. So that's what we are like working very hard on.
Very good. The last question is Professor Vitale, great talk. How well do MCN-based electrodes conform to tissue, and can they be used to study single neurons? And here the question is, would they wrap around the neuron?
Yes, so both the thin film electrodes with like microscale feature size, as well as the larger maxtrodes, they conform really well to the substrate of interest, because perilene, like thin film, can conform really well on wet, exposed brain, heart, or nerve surfaces. And yes, they can also wrap around these curved surfaces. The larger scale electrodes, we make them with elastomers, and we use adhesives, or we engineer and manufacture them in a way that the elastomer retains some type of like adhesiveness. And so because of the elastomer and because of its adhesiveness, they can conform really, really well to the surface of the skin, which is the reason why, for example, we get better performance than regular electrodes in motion artifact rejection. With the wraparound neuron, that is like a very interesting and exciting question to consider. The scale of these devices would not around to wrap them around neurons unless we can, as Dr. Zhang asked, to make them to the nanoscale levels. But they can definitely wrap around very small, for example, nerves, or they can conform really well to very small, like curved brain structures. So, but in the future, it will be very cool to make single cell interfaces.
Okay,
thank you very
great.
much.
Okay, thank you. So I think it's a great time to move on to the panel discussion so that we can have free time to talk about everything here. So about your research further, and about your personal life, like the lockdown situation in like Shanghai as well. So, so I have one question for Xianting. So since you are collaborating with so many people out there all over the world. So how are you holding up with your collaboration in this lockdown situation in Shanghai?
Yeah,
Probably you cannot send your devices, and then all the experimental setup is like paused or stopped for a while. So
right, exactly. COVID-19 is really out of a sudden issue. that breaks down a lot of things. So for the collaborations between our lab and the other people outside China, we usually have two ways of collaboration. The first way is that we really get a lot of data, and to do this data analyze, we need a lot of new algorithm and amongst mathematical tools. So a major group of people we collaborate is mathematicians or biostatisticians outside China. So they have all different fancy algorithms or tools. So that's one way we do collaboration with. So, and the second thing is that have been collaborating with a lot of friends in Europe, like in German and in Switzerland. So we are kind of interested in the mechanism, of a certain disease, like for paralysis or for cancer, whether the mechanism will be different for different nationality. Like Asian people and the Caucasians, they are genetically slightly different. So whether the mechanism of the same disease will be different or not. So that's also something we call a comparative cohort study. So we have been applying for joint research funding with Germany and Switzerland on the basis of that as well. So that's how we collaborate. And also some other labs, they read our work, and they are planning to they already actually sending their PhD students or their postdocs to China, and we work together here offline. So we had a lot of visiting scholars from everywhere, but COVID pandemic kind of stops such collaborations. So that's kind of the three ways we collaborate with outside.
So how are you holding up now with those situations? Have you ever found any positive aspect of finding new collaboration way, like during this period?
I mean, yeah, that's in general, frankly speaking, that's generally really hard. I always believe that human interaction is a very important. Like if we hold meetings offline and talk face to face, things will be much easier to communicate. So frankly speaking, I do not have any a good solution at this moment. And frankly speaking, this COVID-19 pandemic really hurts our lab's collaboration quite a lot. Yeah.
I'm sorry that I asked a very a sad one. I said the question.
No.
Sorry.
I think a lot of people are facing similar issues.
Yeah, right. So especially you guys are working on the bio things, so I think it's more difficult to deal with those situations.
Yes, sure.
Okay. So can you talk about a little bit happier things then? So you guys are the superstars of the ICANX Young Scientist Awards, and then so has it changed any part of your life?
Yeah, I mean, indeed, even before ICANN platform become such popular, me and Alice, we were already friends way back. So I always know that Alice has been working really hard to make ICANN become such a great platform for communication. So ICANX talk is only a small portion of the whole ICANN platform. They also have like a lot of different programs on Tuesday, talks on Friday, and some other things on Thursday. So ICANX has become a very large platform for different people to share their opinion. So I always encourage my students and my my department students to try to follow as much as possible. Indeed, especially for this COVID-19 pandemic. As we just discussed, offline communication becomes harder and harder. So we do need a online, environment to know what's going on, in this world. And I was just listening to Flavia's talk very carefully and take some notes, especially the last question that delivered to Flavia, whether there, this soft electronics can wrap a single neuron. I was thinking that if one day Flavia's lab can make a multiplex electronics that wrap many neurons at the same time, maybe they can stimulate all these neurons and they fix them right after. And then they can send all these neurons to our lab, so we can analyze, using the same electrical power, but at different spatial location to stimulate different neurons at different location of the same brain, how these single cells behave at their proteomic level. See, so this is the immediate, merit of ICANN platform. So I really love this platform a lot. Yeah.
Okay, good to hear. So the same question to Flavia. So since you got this award, has anything changed your life at all, or your research or anything?
Yes. So first of all, it actually got, during the presentation for the award selection process, I had the opportunity to, listen to the talks of all the candidates, and they were fantastic. And I learned a lot. And I got to meet several people, and actually after one of the other awardees is from my same institution but from a different department, Deep Chariwala, and we hadn't had the chance to meet before, and because of the pandemic and all the other issues, and then we actually got a very nice, decided to meet right after ICANX, and got a very nice couple of coffees and very exciting intellectual discussions, which were refreshing. And I also, I'm also advertising the ICANX platform to my students and trainees, because it's a really, excellent platform to disseminate science globally and learn more about very exciting work from extremely prominent experts in the field, right? I think there's like very few places where you can hear so many great people talking about their science.
Yeah, this is becoming a history, I guess. So we can go back to the research questions again. Probably Martin, do you have any questions?
Oh, sure. So first, to our friends and all the 26 million people in Shanghai, we feel your pain, and we believe that the ICANX platform is here to make sure that even though you're in lockdown, we will keep you engaged. So tune in, we will be bringing good science to you. Send us your questions, any comments you have. The platform is meant for building connections where barriers do exist, and we are very happy to help you, but we hope that the pandemic will be done soon. And so coming back to Flavia's comment, it's very interesting that the platform targets to connect people across the world, across the universe, as we say. But here is a case of connecting within the same institution. So it's very interesting that that is also a consequence we're not seeing. And if I can jump back to research, and the organizers of ICANX, the two of you going on stage the same day is very important, because you kind of seem to address two areas that look so different from very far away. Yet there is a point where you both meet. The stimulation, you want to know what you're doing with your stimulation. But Ding is going to map out everything, like he articulated. He's going to map out everything from a single cell. Is there a possibility, like Professor Ding was saying, that we can start thinking about doing electrostimulation, even whole tissue stimulation, and then mapping out the complexity that is resulting from that stimulation, so that we can actually start understanding aspects of neurocommunication that we don't understand. And the reason I'm asking this question is we hear neuromorphic computing coming up as a potentially big thing. The brain is super energy efficient. Our electronics are horrible. Yeah, we like them, but they are not very good. And that's why in 2035, we're going to have a problem. Yet nature has built this system that is so efficient, does the same computational work using a very small amount of energy. Is there any way the two of you or similar different disciplines can come together and help some of us who are not in the biology field understand how we can start building systems that become as efficient as nature. Is it possible? Question to both of you.
So that is like fantastic question. And I think there, a lot we could tell about. It's extremely important to be able to understand how the brain functions. So the brain is extremely efficient, but it's also redundant, right? Like we know that we have billions of like cells, just neurons, right? That they communicate, but we also know that when we have like injuries, like stroke, for example, the brain can reorganize itself. And even if it like some of those like big parts of the brains are lost, the person can perform their function. So there is some level of redundancy and adaptation, right? The way this like mechanisms, communication, redundancy, adaptation work together in synergy to then, perform, like for the brain to perform its function is like so unknown. And it's even like at the more basic level, it's unknown, right? Like when we talk about stimulation, we know that if we stimulate the brain in a person that has Parkinson's or epilepsy, something happens. Seizure stops, the person, if it's like shaking, the tremor stops, but we have no idea of the mechanisms. Like, why does the stimulation work? Like, what is the effect on like local neurons and then all the interconnected circuits so that I can get a therapeutic effect? And so I would love to work with Xianting, right? To unveil at the even more basic mechanistic level, the effects on single neurons and networks of interconnected neurons of brain stimulation. Why do I get a therapeutic effect? What changes, right, in the neurons and in the network of interconnected neurons? And then, yes, absolutely. This, I think, can help us then understanding better how the brain functions in then a healthy state and then replicate that right on our man-made components.
Mm-hmm.
Yeah, so I would like to kind of like address this question from another angle. Our lab has been working with protein quite a lot. In the diagnostic area, they're saying that biomarker. Mother Nature is so great that I think the human being, as far as I know, we have like over 10K different proteins expressed in different organs. 10K proteins is really not a lot compared to how complicated a human being is, right? So 10K protein is not a lot. So how this 10K protein, work together? A small group of this 10K protein like behave like anti-COVID-19. Another group of the same 10K protein like anti-tumor. Another small group, the same 10K protein like empower us to talk to each other, understand each other. So I think this is just by thinking of this is very interesting because the different combination of a not so large pool of proteins, forms different organ, different function of different organ. So that's so beautiful. So I think from me, I think it's really difficult for human beings to completely understand, the Mother Nature's decode methodology. We can only learn from the Mother Nature, but I don't think we can ever completely decode this magic. Yeah.
Very good. I think there's a lot more discussion, and I would encourage all of us to continue thinking about that, because I look at what you're all doing, and Xianting, if I can even take that comment a little bit further, everything is based on an alphabet of 4 letters.
Yeah.
ACTG. The DNA code is 4 letters.
Right.
And it supports life as we know it.
Right.
It differentiates the billions of living things that we know. But yet, when you go to our computation, when we come now to my field in the materials world, and especially in building new electronics, we have such a variety. We have so many elements we can pull from, yet we have not been able to build something as efficient as nature does with 4 elements. So there's a lot of lessons to be learned, and if we can take the basic science of what nature is telling us, transfer it to other fields, and cross-pollinate, I think we will have something amazing. Thank you very much, Miso. Back to you.
Okay, so I was thinking of asking one more question for Xianting. So since you're dealing with so many data, so many number of data, so I'm actually accumulating some data for piezoelectric materials, material properties these days to identify some new piezoelectric material with like innovative properties using like machine learning or something. So during a while, I realized that when we collect some of the data, those data should be classified into like a real true useful data, and then into like false data we have to like garbage data. And then that entails my personal like judgment all the time. So I don't think there's a like some objects that can be some subjective like decision, right? So
Yeah.
while you're doing some research with your data, like a biological data, when you are dealing with those, is there the similar problem, and then how can you deal with that? And then I'd like to have some advice
Right.
from other field.
Yeah. This indeed is, I think, is a common problem. Like especially for big data. Well, no matter whether this big data comes from electronics, comes from biological like proteomics, comes from traffic, from like a city organization. A lot of people saying large data. I think 5 years ago, I was also so fascinated about being able to acquire large data. But I think in the last several years, I started to rethink this issue. Is large data always a good thing or not? I think especially for biological, for medical research, I think the accurate data is more important than big data. A lot of time we acquire so many big data, and then we are drunk in the ocean of data without knowing how to analyze them. But in reality, if we understand which part of this data is indeed the most necessary data, then I think we can spend a much smaller computational power to acquire much higher, like diagnostic efficiency. So that goes back to a very fundamental, like a scientific question. So what part of data actually correlates between the phenomenon and the mechanism? So that is very fundamental basic science, I think.
Okay. Thank you very much. For the interest of time, I would like to present this wonderful certificate for your talks today. Unfortunately, we have to do a virtual one. Xianting, this one is for you for volume 91, and Flavia, this is your certificate for volume 91. And I want to remind everyone that next week, we will have a fantastic speaker, Professor Fulan from Australian National University is one of the co-hosts, and Alice will be moderating that talk. So please join us. You will learn quite a bit about quantum wells and plasmonics. She's a fantastic speaker. And also I want to remind everyone that we have AI Connects Summit coming up on April 26th. And we have lined up a series of speakers, wonderful speakers, Gangshen from MIT, Jagdish, one of the former co-hosts and now president of the Australian National University, and 2 Nobel laureates will be talking alongside some other great speakers for that summit. And with that, I want to say thank you to everyone. See you next week.
Okay.